Welcome to the June RIDE seminar series. Thank you all for joining us in person and on Zoom. For those of you who don't know me, I am Amy Kinsley. I'm an assistant professor in veterinary population medicine, and I'm helping organize the seminar series for the upcoming academic year with the research office and Dr. Caitlin Fiat. I have a few quick announcements before we get started. Our next activity will be the RIDE Summit, and that will take place July 16th. Some emails have gone out with information, but you can also look on the RIDE website. Please RSVP. You can use the link that went out in the emails, or you can also access the RSVP form through the QR code. For today, we'll take questions after each speaker, so please hold them through the presentation. And please complete the attendance form and evaluation sheets. And if you have any suggestions for future speakers, please feel free to reach out to myself, Caitlin, or the research office. With that, I'll hand it over to Dr. Brooke to introduce our first speaker.
I'm Sahar Brooke. I'm one of the first year medicine residents. There's a lot of unfamiliar faces, so I feel very kind of strange to be here, but honored to be. Today I'm going to introduce Dr. Emily Coffey, who is one of our medicine faculty. She is also one of the professors here. She, I believe, completed her residency back in 2018 at the University of Minnesota in small animal internal medicine. She then did a fellowship in immunogenetics, and then did her PhD under Dr. Eva Faro's lab. And then now she's here as a professor. Also, more importantly, she has been a lovely faculty member for us and an amazing mentor. So it's been an absolute pleasure getting to know her over the 1 year that I've been here. And yeah, with that, I want to introduce you guys to her talk.
Thank you, Dr. Should I use a microphone or anything, or we're good? Okay. All right. Yeah, so hi, everybody. I'm really excited to be speaking at the RIDE seminar. I think this is such a great session for the college. As Dr. Brooks said, my name is Emily Coffey, and today I'm going to share some of the work that our group is doing investigating the role of the microbiome in urinary stone disease. So I'm hoping we can have some good, rich discussions, and please ask all of your questions at the end. Perfect. So I want to start by giving you just a little bit of background to get you oriented. So first of all, urolithiasis is a term I'm going to use a lot today. And this is basically just a fancy way of saying urinary stone disease. So we can see calculi or urinary stones that form really anywhere within the urinary tract, moving from the kidneys down through the ureters, bladder, and urethra. And our group in particular studies a specific type of stone called calcium oxalate. So here in the circle, you can see a large calcium oxalate kidney stone. This is a radiograph or an X-ray from a person. And then on the right, you can see some actual photographic images of what these stones can look like in real life. And calcium oxalate stones are very problematic for a lot of reasons. So one of those reasons is that they're painful. And if you think about having sort of this large jagged stone sitting in your kidney or bouncing around in your bladder, you can imagine that might be really uncomfortable. In addition to being painful, these stones are also increasingly prevalent. They're treated by some form of physical removal, which can be variably invasive and can be quite expensive. And then on top of that, even after removal, the stones themselves are highly recurrent. Also, all of these problems that I'm describing are not only true for calcium oxalate urolithiasis in people, but also in dogs. So these stones are one of the most common types of urinary stones we see in both species. And in fact, dogs are a very good model for studying calcium oxalate urolithiasis because they share so many similarities with the human version of the disease. Another problem with these stones is that we know of a lot of risk factors that can put a person or a dog at higher risk for stone formation. So things like diet, medications, genetics, comorbidities, but we also can't use those to perfectly predict who's going to form stones. And we know that there is a lot about stone formation that we still don't understand. And that has really impeded our ability to prevent these stones from forming, hence the high recurrence rates. And so over what I would say is the last 10 years or so in particular, there's been this growing interest in whether the microbiome might represent a missing piece of the puzzle in what is causing calcium oxalate stones to form. Now, in the human world, the bulk of the research looking at the microbiome and stone formation has related to either the gut or the urinary microbiome. And I'll give just a very brief overview of each of these systems and what we know about their relationships to human stone formation first. So if we start with the gut, one of the reasons people first got interested in this question is because there were several studies coming out that basically linked antibiotic use to kidney stone risk in people. And there was a particularly high risk when those antibiotics were administered during childhood. And so this raised the question of why is that association there? And is one possible explanation that those antibiotics are shifting the microbiome in some way that promotes stones to form, particularly when we think about childhood microbiome development. And even getting more focused in on that, people started to wonder about bacteria that degrade oxalate. So oxalate, as you might imagine, is a big part of calcium oxalate stones. And the idea here was that certain bacteria like oxalobacter, which is kind of the main one we think about, would be killed by antibiotics. Then we have less oxalate being degraded in the gut, more oxalate being absorbed from the gut, getting excreted in the kidneys, and kidney stones are forming. So this was sort of the idea behind it. And this idea has been widely studied. So this was a huge area of investigation for a while. The kind of bummer here is that we got really inconsistent results. So people got really excited about it, and some studies supported this theory. But then when we took a step back and looked at that body of literature as a whole, it was actually a little bit underwhelming. And so not surprisingly, it turns out it's a bit more complicated than that. And people had really been focusing on this one bacteria, oxalobacter. And what we're learning now is that it's actually these very complex, highly coordinated oxalate degrading networks that exist in the gut that are influencing this process. Additionally, it isn't just oxalate degradation that's at play here. There are a lot of metabolic pathways in the gut that are being disturbed in stone formation. And one of them that I'm going to come back to over the course of the talk are deficiencies in short-chain fatty acids, which have been observed consistently in a lot of studies looking at the gut microbiome and stone formers. So short-chain fatty acids are basically a large class of metabolites that are produced when gut microbes ferment things like dietary fibers, and they confer a lot of different health benefits. So then just briefly, I also mentioned we look at the urinary microbiome in stone formation. And there's been a lot of really cool work showing how microbes in the urine might influence stones and how they form. So one example of this is that we know many different types of bacteria, if inoculated into urine, will promote calcium oxalate crystallization, aggregation, and growth. So there's a lot of theories as to why. Probably part of it is that they serve as true adhesins that let those crystals come together and cluster. But it's also probably a lot of other reasons too. So we know that the microbes that are in the urine can alter urinary pH levels. They can produce enzymes that can contribute to stone risk. So citrate lyase is a great example. This is an enzyme that can degrade citrate, which is an inhibitor of calcium oxalate crystallization. They can produce or promote the production of inflammatory proteins that get incorporated into the stone matrices, affect metabolism, and all of these other reasons. So with that all said, I kind of want to shift into having set that stage what we are doing here in our research group at the college. And the in a nutshell answer is we're looking at these same questions and these same pathways, but through the lens of a canine model of stone disease. And by doing this, we have a lot of opportunities to advance not only canine healthcare, but because they are such a good model of stone disease, translate this to the human condition as well. And I'm also going to make the argument here that while we know dogs are a good model for calcium oxalate stone formation, I also think they're a really good model for microbiome research in general. When we compare them to other species that are common models, we know that there are a lot of similarities in gut microbiome profiles that exist between dogs and people. Unlike laboratory models, they live in our homes, kiss our faces, drink the same water, have all the same environmental exposures that we do. We have a lot of ability to control for certain variables that are a lot more challenging to control for in human subjects and things like that. So as a good model of both stone disease and for microbiome research, if you buy into what I'm saying, I would argue that dogs are uniquely suited for studying these types of questions. All right, so I'm going to start by sharing some work that I did just so long ago in my PhD. Wasn't that long ago. So this is the first study that we did looking at this question. I was in Dr. Furrow's lab as a graduate student, and we realized we know absolutely nothing about the urinary microbiome in dogs that form stones, despite this sort of growing level of interest in the human world. So we did a very small, just pilot retrospective study using biobanked urine samples. These were from dogs that were all miniature schnauzers, so one of our poster child stone forming breeds, and schnauzers that had stones and that did not have stones, so cases and controls. And it was a little underwhelming at first in the sense that we actually didn't observe any major differences in overall diversity of those microbial communities between groups. So that was okay, but so whenever we look at the microbiome, you'll hear us use terms like alpha diversity and beta diversity. So looking at how diverse the communities are within a sample or between groups. And really, we weren't finding anything super exciting there. They were quite similar. However, when we took a deeper look at the individual microbes that were differentiating those groups, we did find some that were really interesting. And one in particular that I want to make a note of is a bacteria called Acinetobacter. So this was interesting to us because it was the most overrepresented taxa in the urine of our stone formers. And it's also been demonstrated in multiple human studies to be enriched in the urine of human stone formers and in the gut of human stone formers. There's also some cool sort of mechanistic ways where we can create stories about how it might influence stone risk. So this was really interesting to us. But this was a very limited study. So a small sample sizes, a single dog breed, the samples were retrospective, et cetera. And when we see things like Acinetobacter or any other bacteria where we say they're different in between groups, one of the points that's really important to remember is that we can't say that that bacteria is causing stones to form just because it was enriched. We don't know if it was contributing to stone formation, if it was a consequence of the stone being there, if it was a consequence of treatment for stone disease or any of those possibilities. So to better answer that, we really need to characterize what is happening at the metabolic level and functionally within these microbial communities. And that's when we start to get into this omics land, where we can look at different perspectives on how these communities are functioning, using tools like metagenomics or metabolomics and so forth. So the next thing we did is we got a larger population of dogs. This was not breed restricted. We had dogs that had formed or had active calcium oxalate stones. Then we had stone-free controls that were breed age and sex matched. And this time we looked at both the gut or the fecal and urinary microbiome, as well as urinary metabolomics. And unlike the first study, this time we actually saw pretty pronounced differences in the urinary microbiome between groups. So in this figure, the orange triangles represent our stone formers, the purple dots represent our controls. The closer two icons are to one another, the more similar their microbiomes are, is sort of generally how this is interpreted. And when we looked at the urine of the stone formers, it was enriched in Pseudomonas, Lachnoclostridium, and Fusobacterium relative to the controls. Again, a lot of parallels with what we were seeing in terms of what they've seen in the human world. And Pseudomonas was particularly interesting to us because Pseudomonas biofilms have also been shown to promote calcium oxalate crystallization. So just a lot to explore there. We also saw pretty dramatic differences in the urinary metabolomic landscape of these groups. We're still sort of piecing together how this is interacting with the microbes, but we're seeing really dramatic differences in a lot of metabolites, and a lot of those are related to certain amino acid and purine metabolic pathways that, again, have been linked to stone formation. And then finally, we also saw depletions of gut microbial diversity, which is similar to what's seen in people in the dogs that had stones. And that was characterized by deficiencies in a lot of the short chain fatty acid producing bacteria, in particular one called C. or Clostridium hirinones, which is almost become like a biomarker in small animal medicine for good gut health. We know this is one of the truly beneficial bacteria. It plays a lot of roles in bile acid conversion and short chain fatty acid production and so forth. So it was really exciting for us to see that there too. Now, moving just a little bit further along the short chain fatty acid route, we already knew we were kind of interested in this because they've been seeing deficiencies in human stone formers. We're seeing deficiencies in short chain fatty acid producing bacteria in our canine stone formers. And another thing that we discovered is that butyrate, which is a major short chain fatty acid, is deficient when we do serum metabolomics from canine stone formers as well. Additionally, those butyrate levels negatively correlate with oxalate both in the serum and urine. So what that means is the higher the butyrate, the lower the oxalate and vice versa, which kind of supports this idea that maybe these short chain fatty acids are playing a role in what's happening for urinary stone risk factors. And so this has been sort of a shift in our research program where I've really been sort of interested in this short chain fatty acid angle. And when we think about whether these might be protective or not, in the human and the dog world, we know very little about it. But in the rodent world, they've actually done some very cool studies where they showed that if they induced calcium oxalate stone formation in rat models, which they do by giving ethylene glycol, short chain fatty acid administration can mitigate the crystallization effect. So here what you're looking at are histopathology slides from kidneys. You see control rats on the left, EG is ethylene glycol, where there's all these big clear white spaces where calcium oxalate crystals are forming within the kidneys. And then in each of these last three panels, they administered a short chain fatty acid as a supplement. Just visually, you can see they look very similar to the control panels relative to the case panel there. So it is suggestive that there might be some sort of a protective effect of short chain fatty acids in terms of stone formation. And when we think about why this might happen mechanistically, there's a lot of possibilities here. So there's some evidence that it might be altering oxalate homeostasis. So we know that short chain fatty acids have been associated with changes in oxalate transporters in the gut. We know that they can have anti-inflammatory effects within the kidneys that might be protective. They may cause shifts in the microbiome that are protective, or there's some other ideas that have been speculated. Again, though, we don't really know what is happening quite yet. So Dr. Brooke, who introduced me, one of the projects she's working on for her resident research is answering a very simple question we still don't know the answer to, which is, are short chain fatty acids actually deficient in our canine stone formers? We know some of the bugs that produce them are, but we actually don't know the answer to this. So this is something that she's going to be looking at with her resident research, and then she's also going to be looking at how those relate to downstream urinary oxalate and other urinary factors. And a lot of people ask me how this might affect what we're doing clinically. And the short answer is, I don't think we are yet at a stage where we're going to change clinical decision-making because of this microbe being enriched or so forth. But what I will say is that we are currently creating a really robust foundation of knowledge that I'm hoping can be leveraged down the road into clinical applications. And we have a lot of ways we can try to strategically modify the microbiome. We just have to know what we're doing. And that's sort of the body of knowledge that we're trying to build right now. I will give the caveat that a lot of the data in this area, this is a field very much in its infancy, and it's typically small studies with single time points and a lot of limitations. We do have one study going on that I want to highlight just here in the last minute that's called the PICO trial that will try to address some of these limitations and really propel this area forward. So we have our CIC friends here today who I would say are largely responsible for this trial staying afloat. But it's called the prebiotic inulin calcium oxalate trial. That's our catchy name. And here we're looking at how enrichment of short chain fatty acids using prebiotics actually shapes urinary stone risk factors. And it's a crossover study trial design. So basically dogs are put on a standardized diet. They're randomized to get this fiber supplement or a placebo. They have a washout period and get the alternative therapy. We're collecting urine and fecal samples every 2 weeks. And then we're looking at several outcome variables that can help us piece together a picture of urinary stone risk, looking at metagenomics of the gut and of the urine, looking at oxalate homeostasis factors, gene expression of oxalate transporters, and urine biochemistry, so things like calcium and oxalate. So it's a really cool trial that I think will help us move forward in terms of practical clinical application of this type of information. And really, I think this work in general has two major outcomes of interest. I think one, it's going to help us better understand what is causing these stones to form. And that is important in itself because this is an area with a lot of knowledge gaps. And then ultimately, I'm hoping we can harness this again into tangible clinical interventions, biomarker discovery, and things that'll help us improve veterinary medicine and human healthcare at the same time. So, all right, hopefully I didn't rush too much. I think that's all that I have with all sorts of acknowledgement here, and happy to take any questions.
And maybe this will come up in Dr. Brooks' investigation, but dogs are, we think, getting a complete and balanced diet versus people very much, probably not. So how are you reconciling that difference?
Yeah, it's a really good question. And it speaks to, sorry, I will repeat the question for Zoom land.
They can hear.
Oh, I will not. Perfect. So it gets at a larger question too, which is diet in general, which has truly been kind of a nightmare to deal with in these studies.
Yeah.
Because obtaining a dietary history, it's already challenging in humans. And it's one of these things that I sort of tout as an advantage of using dogs is, oh, they eat more consistent diets and we can get better histories. That's not true at all, I've discovered. I get wildly different diet histories from different owners. So we do the best we can, I would say. And typically we do try to get really comprehensive standardized diet histories from every participant. We'll extract data related to macronutrient profiles, fiber sources, fiber content, and try to incorporate that into analyses. What it's absolutely going to have differences that are difficult to account for when translating it to people. But you know, that's sort of it, I guess, sort of a reality of it. But we do try to incorporate that diet angle as much as we can. In the PICO trial, we actually put them all on a standardized diet, which helps us, where the only thing that we're changing is the prebiotic supplement part of it, and it helps us better gauge the effects on this fiber supplement, what it does to short chain fatty acid, how it affects calcium oxalate, et cetera. Yeah, it's a good question.
It's fascinating work. It's very cool. When you think of models, we tend to think of models in the context of present, and we often forget to think about the evolutionary trajectory that got us to where we are. And in the microbiome in particular, it's really interesting because there seems to be a lot of homeostatic plasticity in that when you look at, presumably, so this is an assumption, the way that dogs evolved over the last 90 million years in humans through separate branches, that our microbiomes were probably quite distinct. But there was a really fun study that I'm sure you're aware of in PNAS where they did a neighborhood level analysis of microbiomes. And it turned out that people's and dogs' microbiomes living in the same household were more similar to each other than the people neighbors or the dog neighbors.
Yeah.
So we are exchanging microbiomes with our dogs, and we're having to adapt to these microbiome changes that we didn't really evolve with. So seeing as to how many of the physiological activities that our cells go under don't have that much plasticity, right? The dog immune system is not the same as a person, and a dog GI system is not the same as a person.
Mm-hmm.
What do you think about that homeostatic plasticity of being able to adapt to the bacteria that inhabit us or that make us, and how you can actually extrapolate to disease processes where the physiology may not really be all that similar? And I don't mean to be
Yeah.
it's a philosophical question, and it's not an attack at all. It's
No,
just
no.
a curiosity.
No, it's a really fair question, and I don't have a great answer to it. You know, I think what I would say is I think there's multiple levels when we look at the microbiome. And so part of it is, like, I think the taxonomic profiling angle of it is a really good starting place, but not really all that helpful, because we know there's so much functional redundancy in what these microbes are doing. And I think that's really the bigger piece of it, and probably speaks more to what you're getting at in terms of even when we are forcing changes to adapt within our microbiome, and we're having differences in the disease process and how the host and the microbes interact, the functional component and the redundancy where we can have the same activities happening with different microbes filling those sort of biological niches might be the part that really matters. The challenge, I think, in saying that is teasing through that when you're actually, you know, just extracting DNA and looking at it and sort of trying to understand what is happening at that level and how it's interacting with the host. So again, I don't have a great answer to it. I think it's one way where it's going to be trying to get a more comprehensive look at function and what is happening, what proteins are being produced and metabolites, how we know those interact with disease states in both species, and then probably a level of interpretation that's going to be imperfect. So it's not a great answer, but I think it hits on a key challenge with this type of work, especially doing microbiome work in any species outside of the one of interest. Yeah, it's a good question.
If I share a microbiome with my dog, does that mean I can run really fast and do it?
Well, you know, they've given like FMTs from obese mice into non-obese mice and all those studies. So they do transfer phenotypes. Yeah.
So maybe you should, just following up on that, maybe you should look to see about the humans that own the dogs. Like, do you have
Oh my,
former?
I would love that.
I'm
so
sure
much.
you'd love to do an IRB for that.
Yes, I would need an IRB. I would love that. And I do a lot of urinary microbiome work, and I've always wanted, because they've done the cool studies you're talking about, I've always wanted to look at shared urinary microbes
That'd be really
between
cool.
species. Yeah, especially because we know they can share like antimicrobial resistance genes and things like that. So.
I think you should do it in little kids because they go out on the yard and
Yeah.
Right.
it's just normal behavior.
Sure.
All right, great, thank you. Okay, for our second speaker, I'll invite up Dr. Modiano to give the introduction.
All right, so Amy gave me a text because she didn't want me to ad lib. She also said that a trainee had to introduce her, and I count because I'm a lifelong learner. So Amy Treeful, Dr. Treeful completed her PhD in the CMB program here at the U of M in Dr. Friedenberg's lab. I'm sorry. She studied risk factors for primary hyperadrenocorticism. She's now a postdoc in our group where she's leading research in veterinary immunotherapy trials, and specifically looking at a liquid biopsy for assessing lymphoma risk, and also developing a new line of research investigating how the microbiome influences immunotherapy response in lymphoma trials. So,
Jump stingers. Okay, so now we're going to transition to talking about the fecal microbiome. And I'm excited to share some initial insights from our preliminary data looking on relationships between the fecal microbiome and the response to immunotherapy. Okay, so for those of you who have dogs, I'm guessing that when you take your dog for a walk and you pick up after it, you're not saying, well, there went a potentially valuable clinical test. But maybe you should be, because what's in that bag, the fecal microbiota, could help us solve one of the most pressing questions in oncology right now, which is, why do some patients respond to immunotherapy and others don't? And I don't have my clicker.
It's the other one.
It's this one. Great. Okay, so when we're talking about immunotherapy, more often than not, we're talking about immune checkpoint inhibitors. And research on immune checkpoint inhibitors and their use in dogs lags behind that in humans. So we need to look to the human data to understand sort of the global response rates. And immune checkpoints, represented here, are an important regulatory mechanism for the immune system, and it helps prevent immune cells from attacking and destroying healthy cells during an immune response. And so it occurs between proteins that are on their surface. I'm just gonna use my hand. So you can see here, like for this one here, proteins on a cell, PD-L1, which would be a ligand for a receptor on the immune cell here, PD-1 on a CD8 T cell. And if those interact, they make a handshake that says, hey, I'm your friend, leave me alone, nothing to see here. And cancer cells have gotten really good at co-opting immune checkpoints. And so they display those ligands on their surface to interact with immune cells and to evade killing or engulfing, for example. And so immune checkpoint blockade are therapeutics that are able to interrupt that secret handshake from happening. And they've been very powerful. You see response rates on average of all the patients in humans that have cancer that are indicated for receiving ICIs or immune checkpoint inhibitors, it's about a 20% response rate. But it's context dependent, and you have quite a range of response. So in cancers that have a high response rate, such as melanoma or microsatellite high microsatellite instability high tumors such as colorectal, gastric, and endometrial cancers, you can see as high as 60% of patients responding. But in cancers that have low response rates, you can see as low as 12.5% of patients responding. And those are represented by hematological cancers, prostate, pancreatic, and glioblastoma. And so the remaining question is, how can we predict who's going to be a responder and who's going to be a non-responder? We don't have good tools for that. So right now, some of the tools we have are, as I mentioned, microsatellite high instability, total mutational burden, which incidentally in dogs tends to be a little bit lower, and PD-L1 expression. But we have no uniform, robust tool to be able to determine who's going to be a responder and a non-responder. And as you can imagine, those therapies are expensive. They take the place of something else that could potentially work. And they have side effects. So the fecal microbiome offers an interesting opportunity to develop some more robust biomarkers to identify responders from non-responders. And here I've listed a bunch of different objective outcomes that have been associated with the fecal microbiome. There's varying levels of evidence for each one of these relationships. For example, adverse events is not as strong of a link. These are all still very active areas of research, but immune modulation, Emily talked about short chain fatty acids. For example, short chain fatty acids can induce Tregs. So there's lots of examples here, but probably the most potent example of the relationship is that you can take fecal matter from a patient who is responsive to immunotherapy and transfer it to one who is non-responsive and induce a response. So you can overcome resistance. And that's been shown in sort of three seminal, I'm standing right in front of it, three seminal studies, all done in different countries that showed this in the context of melanoma patients, advanced melanoma patients that were receiving anti-PD-1 therapy. And so, for example, in a study in Israel, they did FMT and showed that in 10 patients, and 3 out of 10 converted to responders. In a study done in the US between NIH and an institute in Pittsburgh, they showed that, I believe it was 6 out of 16 were responders, and 3 of those were complete responders. And then there was another one done in Canada that showed 13 out of 20 patients were responders with a fecal microbiome transplant. So that's pretty powerful. So we're thinking that you can see these relationships, that these relationships would also hold in canine models, oncology models. And so, so we are looking at this relationship in our dogs that are receiving immunotherapy in our immunotherapy trials. And we are specifically asking, hypothesizing that microbiomes with increased and stable diversity characterized by key taxonomic consortia, so a group of microbiomes that move together and stick together, enhance therapeutic efficacy by promoting an inflammatory tumor microenvironment. So moving from a cold tumor to a hot tumor, so one that is not immune infiltrated to one that is. And part of the reason we think that this should be the case is because Emily touched on this a little bit and in the discussion afterwards, but vertebrates have evolved in the presence of microbiomes. So you go all the way back to the dinosaurs. Vertebrates have been evolving in the presence of a microbiome. And also we talked about that study that showed that dogs and humans have similar microbiomes. So there's other research I could cite that also supports that. So how are we going about this? So another thing that we're doing that's kind of unique here is we're actually collecting longitudinal samples. So Emily mentioned that a lot of these are kind of cross-sectional, but from our immunotherapy dogs, we are able to potentially collect a sample at every clinic visit. And so we have longitudinal samples and we can look at the dynamics over time from these dogs. And these numbers here right now are just showing you how many I have in the data set I'm going to show you today. This is an ongoing study and it's exploratory. And I have multiple time points from the immunotherapy dogs. These ones here are cross-sectional. I will add that I have another 3 healthy dogs in the data set that you're going to see that were batch controls for my extractions and sequencing. And Okay, and so this is the workflow. So basically, we collect the fecal samples, do the DNA extraction, use 16S ribosomal RNA gene amplicon sequencing. We're looking at the variable region 4 in the 16S ribosomal RNA gene, and then work on the analysis. I'm also collecting bulk stool samples for potential follow-up later with metaproteomics, so we can look at the functional activity in these dogs. And also I have tumor specimens from, mostly from dogs in this orbit cohort, so we can look at the intratumoral microbiome. Okay, so now we can get into the data. So Emily had talked a little bit about alpha diversity, and alpha diversity is the species richness and evenness within a community or within a dog. And so I have 3 different communities here we're looking at: healthy, lymphoma, and sarcoma. And so we use 2 different measures of alpha diversity here. We use the Shannon index and the CaO1. And the Shannon index looks at both species richness and evenness. And the CaO1 just looks at species richness, but it accounts for rare taxa. So it does a better job of measuring, accounting for some of these more rare taxa. And so what we found was that species richness was highest in the healthy dogs here, and that we did not have a significant result for the chow. So that's basically saying the difference that you see in these different stacked bar charts has more to do with species evenness it's not species, it's at the genera level, taxonomic richness as opposed to, taxonomic evenness as opposed to richness. And so you can see that when you see that these bars have more comparable widths between them than they do in these ones here. Okay, so now we're talking about beta diversity. So that's differences between communities. And so it's the same 3 communities we're looking at: healthy, lymphoma, and sarcoma. And here we see that in 6 of the most common taxa, they had significantly different relative abundances. And if we look at what some of these are, they are potentially biologically plausible. So this is the one that had the most significant difference. That has been associated, and you can see that it's higher in healthy dogs, and most of these differences tend to be higher in the healthy dogs. And in that particular example, higher amounts tend to be associated with a better outcome in lung cancer, and then also reduced amounts tend to make you more prone to some mucosal tumors. And then Fusobacterium, also higher in the healthy dogs. That tends to be a taxa that is more common in dogs, in higher abundance in dogs than in humans. And it's also associated with more outside playtime. Okay, so this is a fun graph. So we looked at co-occurrence networks here. So this is beta diversity. We're looking at community composition, and we compared the different communities that we had. So healthy to lymphoma, healthy to sarcoma, lymphoma to sarcoma. And what we found were that the biggest differences existed between healthy and lymphoma dogs. And so what you're seeing here is not just who's coming to the party. That was measured with the adjusted RAND index here, and it's not significant. So that's telling you that the taxa that are in each community are basically the same. There's no significant difference in who's there, but the influencers at the party are different. And so this degree centrality is telling you which taxa play a bigger role and co-occur with more taxa, or how many other taxa are they connected to. So each one of these circles is a node or a taxa, and the lines are all the connections it has to the other taxa in that community. The thicker the line, the stronger the correlation. The color also resembles the nature of the relationship. So a green line means it's a positive correlation, so they move together. A red line means it's a negative correlation and they move away from each other. And so what we found here, I highlighted 5 on the healthy because you can't see them as well on the lymphoma one, but in the difference between these 2 communities, these 5 taxa become more central. They're connected to more other taxa in this community. And so one way to think about that is if with degree centrality, I mentioned the word influencer, so it's how many other nodes it's connected to. And that means it's more central. It's more of a major player in that community. And this is not a functional analysis, but it kind of infers that there may be some functional shifts that are happening. And then eigenvector is a similar concept, but that is telling you whether those major players are connected to other major players. So are the ones that are having the most influence connected to other ones that are having the most influence? And both of these were significant in terms of the difference between these two different communities. Okay, and this is probably the most exciting preliminary data that we've seen so far. And so this is the principal coordinate analysis, and Emily showed you a similar graph here. And so what you're looking at is how related are the samples within the overall population, the study population. And so it's separating them out using a measure called Bray-Curtis, which is a dissimilarity index, and it has to do with relative abundance. And what it did was separate out two clusters here. And you can see that this big cluster has almost all of the healthy dogs. So it's a healthy-like cluster. And then the small cluster has just one. Also in here are 4 immunotherapy dogs, points from 4 immunotherapy dogs here and 4 immunotherapy dogs here. So the first thing we asked was, are these clusters significantly different? And yes, they are. We had a pretty small p-value here. And then this R value goes from -1 to 1. And being close to 1 tells you that this is a very strong difference. So this is a pretty significant result, even though this is still a relatively small study population size. So then the next question we asked is, can these two clusters be explained by metadata? There we go. And so what we noticed was that the immunotherapy dogs in the healthy-like cluster tended to have less overall adverse events and lower severity of adverse events. So you can see that the highest grade of adverse event that any of them experienced was 2. And total, if we count up all the adverse events of all the dogs, it was 5, and 3 of them had 1 or less. And then over here on the right, we have a total of 16 adverse events, and the highest grade was actually a 4, and it started at a 2. So it's separated out by frequency and severity of adverse events. And caveat, this is early stage, but very intriguing. Okay, so what are the main takeaways from this? As I mentioned, it's preliminary data, but we did see an association with immune adverse events, which suggests that the microbiome could be a potential predictive biomarker to target to modify the immunotherapy response and tolerability to checkpoint inhibitors. And we did not see strong differences between lymphoma and sarcoma. So that kind of hints that maybe there's a non-specific malignant microbiota signature. And we saw stronger differences between the lymphoma samples and the healthy samples as opposed to the sarcoma and the healthy, suggesting that maybe lymphoma is, we'll see a bigger difference if we, that lymphoma could be a more impactful target than sarcoma. And I kind of hinted at this. The network shifts with stable taxonomic richness, so it had more to do with changes in the evenness, is suggestive of functional reorganization. So future and ongoing work, we're doing ongoing sample collection. So these are all ongoing studies and we're continuing to collect samples from them. And we're also going to broaden the study cohort to improve detection of a specific lymphoma or malignant signature. And I'm looking to do intratumoral microbiota analysis. That's kind of in progress. We're banking the bulk stool for potential functional analysis. And One thing that we're going to look at probably when we have this next batch of samples processed is looking at the longitudinal principal component analysis and longitudinal networking. So doing that same networking analysis, but with the different PCOA clusters to see if they have different major players responsible for those differences. And then we also have a lot of other data in these studies that we can integrate with, such as spatial genomics, single cell RNA-seq, attack-seq, and bulk RNA-seq. And that will allow us to see if some of these changes and these signatures associate with some immune functionality. And then we are going to also extend the integration with metadata. So looking at diet, we have diet information for all of the dogs in our study as well. We also have follow-up, every 6 months, we have follow-up information for all of the healthy dogs. Okay, and there's a lot of people I have to thank for being able to get this research done. The CIC really helps me a lot getting my samples. And this is a collaboration. The immunotherapy trials are a collaboration between the Mayo Clinic and here. And then this is the fuzzy weirdo that I share my microbiome with. Yeah, Eva.
I'll ask a question.
Okay.
With the time, I know you said you were getting multiple time points, which I think is going to be really cool to look at when you have a larger sample size. With the ones that you have so far, where you were showing kind of the clusters in the group with differences in adverse events, do you know if the samples were collected before or after the adverse events had occurred? Just
Right, we have
when I
both.
think of adverse events, I'm like, if they're GI related,
Yeah.
they're gonna have a diet change, they're gonna end up on probiotics,
Right.
like all these other things are gonna happen.
Yep,
Yeah.
so we have all of that information and so we'll have to go back and look at the exact time and date, but all of those events are put into REDCap and we have it. So we do have pre-treatment and post-treatment in the same analysis cohort. So you're seeing all the different time points for a dog in that analysis, and they are happening at different times. So I don't have it actually mapped out to which one is at what time yet, but we have that information and we will look at it.
Amy, so
Yeah.
what level are you looking at circulating metabolites?
So we're not there yet, but that's what the bulk stool is going to help us look at potentially, and not circulating because it's in the stool, but it could potentially, we do have serum from these dogs as part of these studies. So it could, if we have leftover, we could potentially look at that in correlation with the metabolites in the fecal microbiota as well. But yeah, that's not something we've been looking at or have.
Because ultimately that sets the connection, right? It's gotta be, it's
Yeah.
gotta get to the tumor. You're not talking about
Right.
gastric tumors here.
Well, yeah, so what's happening, so yes, and a good example of that is menaquinone. Plasma levels are actually correlated with a lack of immune adverse events. And so yes, definitely circulating plasma proteins would help us figure that out. But if we look at the metabolome of the fecal microbiota, it can help us see what's being produced there, and then you can kind of infer that some of that would be in circulation, but being able to follow it up with actual plasma samples would obviously be the way to go.
There's a Zoom question.
Okay.
What role do parasites potentially play in samples?
That's a very good question, and I don't know the answer to that. So, Emily, do you have any idea?
I don't have a great answer aside from, I know there are some studies showing the effects of like dewormers and antiparasitics on the microbiome. And I would imagine, I suppose, that if a dog had a true pathogenic infection with something, that would very likely influence the microbiome, but beyond that, I don't have any great insights.
All the dogs in the clinical trial have to be on parasiticides because they are getting vesicular stomatitis virus, and so we have to make sure that we mitigate the risk of a fly biting the dog and transmitting the VSV to a sheep or something. So they all get, I don't know, one of the modern thingy-majigs that you internists use. And I think we have a question for the dogs in Lyra, correct? We actually asked
About
them.
medications,
Yeah,
yes.
so I would say that with pretty good certainty that all of the dogs in that study have or will be on preventatives. So I think it's a really good question, but probably one that we can't answer because we won't have data separating ones that have parasites.
Do the healthy dogs get that as well?
Well, the healthy dogs are coming to us with whatever the owners are giving them, but virtually all of them are on Rebecto or, whatever those things are. So, something that I give my dogs every month and I don't.
Yes, I'll make a fun comment,
Okay.
which is that the beautiful dog you're seeing on the screen was one of the data points that I showed you. Urinary microbiome.
He's got a lot of uses. He's a data point in mine as well.
I don't know if that's ethical.
Yeah.
Give consent.
See the informed consent.
There was actually a comment there. There was a paper published which cited our studies as a major evil doer because we are having clinical interventions of dogs where the dogs do not get to consent. So the owners consent, but the dogs don't consent. And so this was from a philosopher. And he was asking where the boundaries of ethics are and whether it's ethical for us to do these clinical manipulations in companion animals that don't have a choice. So, rather than just be dismissive and say, well, what kind of a crazy person is that? It really is food for thought about where do we draw our ethical boundaries. And the things that we do today or that we don't do today that would have been thought of as crazy or would have been perfectly acceptable 10 or 20 or 50 years ago. So I think it's
Give them some treats.
Yeah, yeah, right.
Yeah, I was gonna say, my dog always consents to taking a pill in a pill pocket. We had someone talk from pediatrics about a similar, children also don't get
Yes, the
consent.
dog that
They're
doesn't
similar.
sign has a need.
All right, thank you both to our wonderful speakers. Don't forget to RSVP for the Ride Summit.
Great job.
Yeah.
Yeah.